the director of MIPSI. Gives me great pleasure to introduce Professor Laroussi from Old Dominion University to give today's seminar. Munir received his PhD in electrical engineering from the University of Tennessee at Knoxville, and now is the director of Old Dominion University's Laser and Plasma Engineering Institute. Munir has been a leader in the field of atmospheric pressure plasmas and their technologies since the early 1990s. He is the inventor of the uniform glow discharge at atmospheric pressure and is likely the inventor of the use of such plasmas for sterilization and biocidal purposes, holding the first patent in that area. Munir's interests in low-temperature plasmas are very broad. It generally covers whatever is a non-equilibrium discharge he is interested in and has made landmark contributions. His most recent work and the title of his talk, and it's the topic of his talk today, is on biomedical applications of non-thermal plasmas. As I mentioned, he started off with investigating the germicidal aspects of these plasmas, but recently has been looking at the applications in dental areas, in wound healing, and in cancer therapy. In Munir's career, he has well over 100 publications in journals and conference proceedings, holds several patents. He has provided tremendous service to the field, having served on the administrative committee of the Nuclear and Plasma Science Society of IEEE. He is a fellow of IEEE. He was the general chair of the International Conference on Plasma Science in 2010. He serves on the editorial boards of Plasma Processes and Polymers, the International Journal on Plasma Medicine. In recognition for his pioneering contributions to the use of plasmas in biomedical areas, he received the 2012 Nuclear and Plasma Science Society Merit Award, which is the highest award given by that society. In addition to that, he's just plain a nice guy that everybody wants to collaborate with and gives great seminars. The title of Munir's talk is Plasma Medicine, Low-Temperature Plasma as a Transformational Technology for the Healthcare Field. But before he starts, he receives, the only reason why he came here today is to receive the MIPSI mug. So MIPSI on one side, University of Michigan on the other. Munir, thank you for coming.
Thank you. Well, thank you, Mark, for this nice introduction. I'm a little embarrassed, actually. He said too many good things about me. That it was way too much. But thanks, I appreciate that. I come from Old Dominion University, where the weather is warm, the beach is not far, and science is flourishing. So I know that here Michigan is a much colder place, but I see a lot of very, very eager people and excellent faculty and scientists that you have here. And I'm very, very privileged to be here talking to you and being invited by, actually, Mark is really a leader in our field. So thank you again for this honor to come and talk to you guys. My talk is on low temperature plasmas for biomedical application, basically. I had to choose and select which topic I will cover because through the years we did many things on the biomedical, so I had to leave some stuff off. For example, I won't be talking about dental application and things like that. I try to cover, what most people are talking about today, and when you go to conferences and meetings that talk about the biomedical application. But before that, of course, I'll show you a little bit of our plasma sources and devices before I talk about the biomedical application. So the work that I'll show you, obviously, there's a whole bunch of people who contributed to it. The students, most important, of course, and then faculty members and postdocs. So there is a whole, it's really, the credit goes to a lot of these people. I just happen to be the lucky one who happened to work with them. So the outline of my talk is I will talk about low temperature plasma sources and show you some of our bacterial inactivation, destruction of some proteins that form amyloid fibrils, cancer treatment, effect on some skin cells, two types, fibroblasts and epithelial cells. And then I'll end up discussing a little bit what we don't know, which is the fundamental, the basic science behind what's going on, which is the subcellular and molecular mechanisms of interaction of plasma with cells. And then I'll conclude. So if you come in here to listen to my talk thinking that I'm going to give you great answers, you will be somehow disappointed, because I'm going to start showing you the questions I asked myself when I first started working on this, and I'm going to end up showing you about the questions I'm asking today. So there's going to be a lot of questions, very few answers, unfortunately, but asking the right questions is half the battle, I think. So if you ask the right specific question, you're already halfway there. And I think in this field, at this moment in time, we are really trying to formulate the right questions. So these are cold plasma sources at our lab that we developed through the years. As Mark mentioned, we first developed this large volume. I think he gave me this laser. All right. The large volume DVD discharges are, everybody uses that concept more or less. We back in the late 90s, we started drifting from going from sine wave excitation to pulsed, because there was a program that was actually funded by the Air Force, and I was very lucky to be part of that, where they were looking to how to generate dense plasmas for different applications. At atmospheric pressure, with densities of electron 10 to the 13 and above in air. And the result of all that work was a big consortium from Stanford to Princeton to Tennessee at the time I was there, etc., where the consensus was using pulsed will get the electron energy distribution function to shift to the higher energy, and get that will lead you later on to a much enhanced chemistry in your plasma. So we started using this short pulses. We also developed, with my professor, we developed this source. Which, my professor's name is Igor Alexev, and we developed this, what we call, this is like the DBD, we called it the RBD because it's a resistive barrier discharge instead of a dielectric barrier discharge. And the advantage, by the way, this is wrong, I don't know what happened, this should be hertz, 50 to 60 hertz, not kilohertz, where we can actually use all the way, instead of sine waves, we can, at kilohertz, so we can use, plug it straight into the outlet, or use just straight DC, and we can generate some nice plasmas. And then when my work progressed more and more into this biomedical application, I wanted to have the plasma out. I don't want it to be confined between two electrodes like that. I want the plasma out. So the idea was to generate a plume, a jet that actually comes out into ambient environment in room air. And we developed what's called the plasma pencil first, and we had different designs of all kinds to get a jet out. The main thing is for biomedical application, the temperature has to be low, 40 degrees or less Celsius. So these jets are all biocompatible, if you want. This is my hand here touching it, and the temperature is low. And we did, of course, all the measurements necessary to verify all that. We also discovered that those jets actually, we used ICCD, are made out of very fast, what we call, these are small plasma packets, and we call them plasma bullets because it sounded good, you know, a bullet. It was a postdoc working in my lab that suggested they call them a plasma bullet. It was not my idea. I have a very short story about that. I was attending a talk by the group, actually from Grimmy in France, and the guy said, the word bullet is wrong. We should not call them plasma bullets. I don't know why people call them plasma bullets. So in our group, we call them plasma streams. And our device is called a plasma gun. I was like, so you have a plasma gun, it doesn't shoot bullets. So make up your mind, either. So anyway, so they had the plasma gun, shoots streams. I suggested, maybe you should call it a plasma hose or something. At least it will make sense. So anyway, the plasma bullet is misleading because this is really an ionization wave. That's a part. It's not a volume of matter that's traveling at this, because these things, they travel at very high velocities. It's supersonic, so they are very fast. We did that back in 2005, 2006. I believe I have a short movie here that shows you so this is what you see with your naked eye, and this is actually what's happening. The tube stops here, and then you have this bullet getting shot, and it goes and then collapses and then dies out. So the discharge is inside the tube, which is here. I'll maybe play it twice. So the tube is here, and the discharge is ignited, and then whoop, this thing is shot right out of that. So this device really shows a train of fast-moving plasma ionization waves that will look like plasma bullets. And we did, of course, all the necessary measurements on this using, spectroscopy and fast photography and what have you, to try to at least understand what on an experimental level this bullet and their structure and their velocities. And I'm not going to show that. That's a whole talk. It takes about the same amount as this whole talk. So we did this measurement. We discovered, for example, the bullets are donut shaped. They are actually hollow. They come out hollow and then they collapse. But that's a whole a new topic. I'll show you very quickly. The taking head-on pictures. The bullet is actually hollow, and then it goes on as time progresses, and then it collapses. And we published many papers, and we also looked at it spectroscopically, and we did Abel inversion, and you can see the edges are luminous, and then it's hollow in the middle. And as it moves, away, 4 centimeters, for example, it becomes more, it collapses, and it becomes, it collapses on itself and no longer hollow. We can also put them in arrays or like this, and as I mentioned before, they are cold enough to the touch and electrically safe, and that's very, of course, important. If you want to use them for a medical application, they need to be cold, and you can't electrocute the patient, right? I'll skip that. We also developed what we call micro pencils. So this is our plasma pencil, and this is a tube, and the bullet is sent down the tube and then exits with a small radius or diameter. So these are really driven toward the people who do the dental application. So they can introduce that into root canal and what have you. And that's I decided, skip the dental work for this talk because that's also a whole topic on its own. I have a student who just completed his master's degree where we I just want to show you very quickly what he did. This is a device that generated jet, but instead of in room air, do it in a controlled environment, in a chamber where we control the contents and control the pressure. And you can see how the jet gets longer and longer, up to about 28 centimeter as the temperature, the pressure goes down, is lowered. But then as you go below 75 torr, other phenomena starts to take over, and actually the jet becomes shorter and shorter, and then we ignite a large volume into the chamber, which I think I have a time-lapse movie here. You can see here as the pressure goes down, the jet is getting longer, and then when you hit that 75 torr, it goes down, and then at low pressure, about 3 or 4 torr, you ignite a large volume plasma into the chamber. This has some application that we are thinking about now, which I'll discuss maybe if Mark invites me a year or two from now. I'll come and talk to you about some of the application of this. But this study allowed us to understand the importance of course, the environment where the plume is launched into, and also the importance of a boundary layer around the helium channel, etc. And as I said, I hate to kind of tease you, but this is a really very long story that, and I'd like to kind of dedicate my time more to some of the biomedical application. But there is a lot of work that is being done by either our group or other groups on these plasma jets and plasma bullet to try to understand their physics and chemistry and what have you. Again, we did the measurement of the temperature either by measuring the temperature directly or by spectroscopy, and of course by touch, etc. So we know they are safe to be applied to a tissue, for example, and we analyze their spectra. We are interested in what species they generate, chemical species such as OH or atomic oxygen that have biological implication. Okay, biomedical application. So I kind of skipping all the plasma things here, going into the biomedical application. The scientific objective is really to understand the fundamentals of the interaction of plasma with both prokaryotic cells like bacteria and eukaryotic cells like mammalian cells. And because that knowledge, unfortunately, is still lacking. The grand objective is actually to use the plasma to overcome unmet medical and healthcare challenges. So what are the unmet challenges I'm talking about? Well, there's as simple as, for example, food contamination. Every day, not every day, but every week, you read about some sort of outbreak of food poisoning. If I'm sure you heard about, even in the last few months, there's a couple of incidents. for, Listeria and Salmonella and E. coli and all that. And you would wonder, with all the, the sterilization and decontamination method that are available today, why we still have these challenges are still around. And more unmet challenges, drug-resistant bacteria is a big problem. For example, MRSA, from 1999 to 2005, the MRSA-related hospitalization is more than doubled. And in the United States alone, you have 90,000 patients per year are killed or die because of infection from drug-resistant bacteria such as MRSA, and they get contracted in hospitals. Chronic wounds. There are wounds that chronic wounds are wounds that persist and don't heal or never heal. And for example, diabetic ulcers, and there are thousands of amputations per year in the U.S. alone because of this wound. And so the healthcare field until today, we're still lacking into methods and therapies how to deal with these chronic wounds. I show you here, I borrowed this actually from a German friend of mine who, you can see here the number of registered antibiotics are going down, while these are examples of bacteria that are developing resistance to the antibiotics we have today. So this is a really, a real and present crisis and present danger that we have. And that is also, alarm bells are going in because of that. For example, I found this not long ago. I have 8-country study finds high rates of drug resistance. And so this is a real problem. The bacteria have learned how to deal with antibiotics that are available today, and they are developing resistance to it, and we are losing the battle, and it's only getting worse. This is, those are kind of in news type, but this is, these are papers from actual scientific journals. The urgent need for new antibacterial agents, the crisis of new antibiotics, what is the way forward? So this is the medical field, raising the red flag, saying there's a problem. We need new technology. We need a new approach to this problem. So can low-temperature plasma be the answer? Why not? This is a new technology that we are working with that have showed some potential in killing bacteria of all kinds. And until today, bacteria, nobody has reported any resistance that bacteria can build against plasmas. Now, what kind of plasma do you use? Obviously, you cannot use all kind of plasmas. Here I'm showing you on top this kind. None of them is suitable for biomedical application. So back, as Mark said earlier, back in the early 90s, there was a whole field that, or subfield, if you like, where people really, it was kind of a hot field at the time, people developing low temperature plasmas at atmospheric pressure. And that made it possible to actually go into biomedical application. And I'm showing you here a few examples from Eindhoven and Drexel and our own lab. So what are the active agents in a plasma? Well, obviously, temperature could play a role, thermal effects, UV radiation, although UV is something that should be actually for medical application minimized, because we know the effect of UV on DNA, and you would like to generate a plasma without UV, and charged particles, and of course the reactive species, oxygen, based reactive species, nitrogen-based reactive species. Of course, Mark and his team know all about the reactions that happen and how these reactive species are generated in a plasma. These are but a few. You have hundreds of reactions, especially when you use air. But the species we need is the reactive oxygen species, nitrogen reactive species. So these are the ones that will have real biological implications. Here, for example, reactive oxygen species, these are a few examples, and their biological impact is like peroxidation of lipids, and the oxidation of proteins, of DNA, and the nitrogen reactive species, for example, NO, is very known. There is a whole field, for example, there is a whole journal called nitric oxide, NO. That's true. There's a journal called nitric oxide because of the biological and medical effect that have been studied with NO. One of them is cell signaling, is initiating signals into cells. So plasma generates a cocktail of chemical agent with biological implication. So that much we know. Okay. Okay, I think this is a repeat of what I just said. So I will move on. And we can measure this reactive. For example, this is in my lab. We measure the ozone we generate from an air plasma. We measure the NO2 under dry air condition, humid air condition, and you can measure, we have probes to measure NO and to measure all this. So see, these are, we can actually measure absolute concentration and fluxes of these species, so we know what kind of environment the cells are being subjected to. Some time ago, I collaborated with two really bright people from University of California, San Diego. When we did a little bit, we published the paper in New Journal of Physics, also on charged particle and their effects. Now we only looked at the physical effects, not the chemical effects. And now, if you model your cell here by, you can model anything by a sphere. So this is let's say a dielectric sphere, which is your cell, and then you ignite the plasma. And of course, the electrons, which are easy, they move easily, etc. They can charge the outer membrane, and then there is this outward electrostatic force that suddenly develop and try to tear that membrane apart. So the calculation showed that force goes as the inverse of the square of the radius of curvature. So if you have cells, and this is a little of an exaggeration here, that have irregular surfaces, so with a small radius of curvature, then that force becomes really large and can tear off part of the membrane, and then the inside starts leaking out. And we observed that actually in our experiments. However, I just wanted to show you this, so I'm not only talking about the reactive species, but there are physical forces. Now, this condition will happen if the plasma is actually directly in contact with the cells. If there is, liquid around that, then this situation becomes less likely. So anyway, what the calculation was that this is the potential, the charging potential on the cell. If it gets greater than this here, this is the tensile strength of the membrane, then you get you get disruption is more likely to occur at surface irregularities. Now, the way to expose a biological material to plasma, it can be there are two methods. One is called direct exposure, where you put the actual sample in the plasma, and the other one is called remote exposure or indirect exposure, where you put your sample at a different, not in the plasma, but at a different chamber or away from the plasma. And in this case, of course, charged particles were usually recombined before getting there, and of course the temperature effect would be minimal. Reactive species with short lifetime will already recombine before getting there, so it will be mostly long-lived reactive species that will get to the sample, versus here the sample is subjected to everything. Okay, now I'm going to go through all this, so, with the bacteria and then the cancer cells and all that. So I thought I'd show you a little bit, very quickly, a timeline of my research activity in this. The first experiment that I did was back in 94, 95, and then we published that in 96, and that was basically just exposing or treating bacteria with atmospheric pressure plasmas of this kind, and we found that it can kill those bacteria, and that's where we got excited and we said, oh, we got to go and do more. So we started studying the morphological study and biological pathways, etc. And then we developed the plasma pencil that shoots this plume. And then we started doing cytotoxicity and wound healing and destruction of proteins. And today we are really concentrating a lot of our work on cancer studies. So I expect in the next few years we'll be doing a lot of this. This is a paper that was published in '96, and the reason I put it there because I'm going to start, by showing you what are the questions I asked back then, and I'm going to end this talk asking just better questions. And that's really what all this work, all these years only get me to. So not too many answers, but just to shape a little bit my questions. And so you can see from back then, when I really didn't know much about this, so I was asking, which plasma regime will be more effective, what is the minimum plasma power density that I need, which physical processes dominate the process, what biological chemical processes induce the death of the cells because I was killing bacteria, which type of gas is more desirable for a particular application. A lot of these questions are still not completely answered, and that is, more than 15 years ago. More than 15 years have passed, and we have only a few answers. I guess science is difficult, right? It's not so easy. So I show you bacterial inactivation. We tried all kind of stuff. This is I don't know if everybody knows about biology here, but very quickly there is a bacteria type called gram-negative and gram-positive. The gram-negative, here I give you some examples, which we tried with our plasma, and these are usually bacteria that don't. They are not able to develop spores. Usually bacteria usually gram-positive bacteria, on the other hand, they can develop spores because when the condition gets harsh, they can go into this dormant state, and they can survive in that state for a long time under very, very tough conditions. The gram-negative, they cannot do that. Now, the positive and negative, this is a stain, gram stain, and that's those words come from. But the implication are important. This here, the gram negative. Here is the gram negative has this. This is the outer wall or membrane. They are very complicated actually. There are layers of they call it peptoglycan layer, and it's very thin for gram negative. But gram negative also has an outer membrane and inner membrane, with a lipid bilayer and some proteins embedded. Now this is the wall that protects the inside from the outside. So, and it has gates that can open to let nutrients in, you get molecules in and out. But it has to be in a controlled fashion because the cell needs to keep what's inside, what's outside, outside. And so then this is the wall with some gates, of course. For gram-positive, you have the peptoglycan layer, which is very thick, and an inner membrane that resembles that. So structurally, they are a little different. And as I mentioned before, the gram-positive bacteria, they are able to go into this dormant state, which they say they sporulate. They become spores, and they can survive. So when you develop a method to kill bacteria, for example, you always have to test it against spores because that shows that your method is efficient in killing that type of those bacteria that are the most resistant. The gram-negative is a lot easier to kill them because they don't have that ability. The early work that we did in the 90s showed, in E. coli that in a matter of few minutes we can reduce their number greatly. This is Pseudomonas. This bacteria can form biofilms, so they are very important. And we showed that we can kill them also, but a lot harder. This is gram-negative, gram-positive, I believe. Later on, when we developed the plasma pencil, we also checked what the effect of this plume, which is a little different than the other plasma, on bacteria. So these I'll show you a few examples. This is a control, and then we apply the plume in a center like this. So this is a region of inactivation. And then, of course, this for 30 seconds. For 120 seconds, the effect is much more pronounced. And also, if you use only helium, this is the zone of inactivation. If we add a little bit of oxygen, this is O2, if you cannot see it, for the same exposure time, you get a much better effect. So we were just testing the efficacy of the plasma plume on cells. I'll show you other examples. This pneumonia and Vibrio cholera, etc. And you can see the effect also is different from one type of bacteria to another. So you cannot generalize. You cannot do a test on E. coli and assume that's going to work for everything. Undiluted, this is, I believe, the concentration was 10 to the 9 per milliliter. And then this is 10 to the 7 because we diluted 100 times. And you can see also when you start with very high concentrated or high concentration of bacteria, the effect is localized. If the initial concentration is lower, then you get a much larger area of effect. So it depends on the type of bacteria, on the initial concentration, which they call the bioburden. Depends on also the substrate on which those bacteria are seeded. So it's a very complicated process, really. There's many factors that will affect the efficacy of the treatment. This is some SEM micrograph showing you the control cells versus the ones that have been treated. This is E. coli, and this is where we see that disruption of membrane and leakage of the cytoplasm, and you get a big mass after you treat them with plasma. This is spores. This actually work that I did with Joe Birmingham. This is a company that was interested in this work, so we had did some tests together, and this is using some spores, and these are the control, and you see the spores have been greatly damaged. This is lysed, basically gutted out, and this was using an air plasma, not a helium plasma. Okay. I started a collaboration with the biochemistry department in our university, where there is a biochemist that her specialty is in this particular protein that form what's called amyloid fibrils, and these are involved in for diseases, neurodegenerative diseases like Alzheimer and Parkinson, and there is no other way to destroy these fibrils. And she heard about our work on plasma, wanted to see if plasma can actually break this fibrils. Okay, so this is a model showing you what they call the beta sheets, which are stacked, well, there are several models. This is one of them, the beta sheets they pack in a stacking manner and form this elongated polymer and form this really stable structure. And those can develop for example, in people who need dialysis, and you can develop this type of protein. This is a different one here I'm showing you, that goes into the joint and causes a lot of pain and restriction of movement, etc. So these are proteins, okay, that can actually stack together and form this network of fibers, and it's almost like plaque that will, if it's in the brain, it would be like a layer of plaque, or in the joints. In the brain, usually there is a region in the brain called the substantia nigra, that where it has the neuron that can generate dopamine, and if this plaque develops and kills this neuron, then you have lack of dopamine production, and then that results in the symptom of Parkinson, which is the rigidity of the muscle and the tremors and all what probably you all know what Parkinson's disease is. So we tried with our plasma jet to treat these fibrils, and I'm going very fast here, so I'm just showing you results. This is how these fibrils look like. This is the network of this particular one are called alpha-synuclein, and they are the ones who are associated with Parkinson, not Alzheimer, Parkinson disease. And you see here 6 minutes, and you see substantial breakage and damage to the network of this protein, which is a good sign. I'm showing you here 6 minutes. However, we do see breakage starting at 2 minutes, and at 10 minutes we basically obliterate all of them. But I'm showing you the case where some of them are still somehow in a fibril stage, but a lot of them have been cut to smaller units and have been damaged. This study is still ongoing because we need to, for example, to see if these small units is there any virility associated with them, and we need to see also the damage the plasma can do to neurons, because you don't want to damage the neurons while you are obliterating this plaque. So still many questions unanswered. Sorry, this is a changing topic now. I'm going to some of our results on cancer cells. We worked with leukemia, this T-cell leukemia cells, and same thing. We are using our plasma jet, our plasma pencil, to treat this. Leukemia is caused by overproduction of these white cells, and white cells are usually made in our bone marrow, and then they go into our blood, and the body regulates all that. However, when a person contracts leukemia, you have this overproduction of these white cells, and our question is, are we able to destroy this, the cancer's leukemia cells? So here I'm only showing you, a very quick example. Show you how in about 3 to 4 minutes we get substantial kill and reduction, and this was measured 12 hours after treatment. So we treat at different times, of course, and then we wait 12 hours and look at the viability of the cells, and then plot this out. And however, we follow this at a longer time, and we see what happens later. I'll show you that in a minute. The method that we use is called the trypan blue exclusion assay. This is a dye that you put in there, and then the dead cells, because their membrane has been destroyed, they will let the trypan blue in, and then they will look like a dark blue dots. So this is the untreated cells, and these are 2 minutes treated. I selected this because I want to show you. See, this is a cell that's still okay, but this one I've taken the trypan blue. These are okay. This is 10 minutes, and you'll see that most of them I've taken the trypan blue, and we practically killed more than 90% of the cells. I thought I had, I'm sorry, I was going to show you 12 hours, 48 hours, 60 hours, but I might have skipped that. I apologize about it. Because as for 12 hours, we stop somewhere here. But if I was going to show you 24 hours later, and then I believe was 70 something hours later, this here will go all the way down to here. So we get a major killing of the leukemia cells. This is another cancer study, which is on metastatic prostate cancer from a site in the brain. This is treated by our plasma jet, and this is right after the treatment. So kind of 0 hour after treatment, we look at the cells, and they don't look like nothing happened to them. They're just fine. We haven't been able to kill them, and it shows. We do the count, and there's not much change. However, 24 hours later and 48 hours later, we keep following, and we see cell dying. This usually an indication of cells can die 2 ways. One is called necrosis, and the other one is called apoptosis. Necrosis is when the cells is kind of blasted away, and the content usually leak out, and necrosis causes inflammation. That's why we have inflammation, for example, when we have wounds, because our white cells will attack the infection and try to cause to kill the invasion of the invading bacteria, and there's a lot of necrosis that happen, and that causes the inflammations. But apoptosis is called programmed cell death, which the cell, when it dies, it keeps within its a sac, if you like, and that's normally the natural way for our cells, because we have our cells die all the time, right? Our cells die all the time and get replaced, and usually that's a programmed, and they call it programmed cell death. And there is a whole cascade of events that happen that tell the cell, hey, it's time to die, and there is a process to be followed, and the cell will die, and our body will get rid of it. And this happens in natural, normal cells. So cancer cells are cells that actually forgot how to die. and which means that cascade is interrupted somehow. And they keep replicating and replicating and replicating. They become what's called immortal, because they keep going and going. So usually when apoptosis happens, there is a delay. And so this delay here of cell dying could be a sign that we are inducing apoptosis in the prostate cancer cells. I show you a picture. These are the untreated cells. These are the 10-minute treatment, and you can see a lot of dead cells down on this corner of the picture there. Now, there are some assays that need to be done to verify if it's really apoptosis that's going on, and we are working on those, so I don't have data to show you on those assays. But that's important. That's something that needs to be done to confirm that the cells are dying by apoptosis, which is the programmed cell death. We also did some work on healthy, mammalian cells, and that is related to wound healing. A lot of my colleagues give presentation and they show you, wounds and burn and all that, and treat it later by treated by plasma and later got better, etc. I have not done any of these studies, so I don't like to show those because I don't know if I feel comfortable showing something that I haven't done. So in our work, we try, always work on the cellular level and try to actually understand at least what's going on the cell or subcellular level, not on a whole muscle or a whole tissue. We have not done any human studies or anything, but this here is related to that. It's because for wound healing, you have to study what are the effects of plasma on cells that are involved in the wound healing process. So we did. This is our protocol, basically, that was followed by the biologist in doing this work here. So we selected two cell lines. One is called, these are all kind of skin type cell line. One is called the epithelial cells, and the other is called fibroblasts, which are, as I said, involved in wound healing. And we want to see the effect, because for sure you don't want to apply plasma if it's going to make the effect worse or you mutate the cells or something bad happens. So we feel very strongly that fundamental studies on the cellular level needs to be done first. So these are the two cell lines I'm going to show you today. This is the fibroblast. You can see a little bit of a description of these cells here, and the growth media where we grow them, and the epithelial cells, and that's how they look like. And this is the type of cells. Adherent means they attach themselves. So you have to have protocol how to get them to out so you can count them, etc. I think these are non-adherent. No, they are adherent too. The fibroblast. So and the growth media where we grow them. So our goal was how much plasma can we apply to these. These are healthy cells, not cancer cells or anything like that. So how long can we apply the plasma before we start damaging these cells? Because for wound healing, that's a very important step, as I mentioned. So the first line, I can show you here pictures, the untreated, and then here 10-second treatment with plasma, and we look at them 12 hours later, and 30-second, and we look at them 12 hours later. Up to 30-second, the situation still looks good. There's a lot of healthy cells. It looks more or less like the control. So up to 30-second, plasma exposure appear not to cause too much damage in this fibroblast. Fibroblasts it's very important for their proliferation, so the wound can close and heal. So 30 seconds is okay. However, when we went to a minute and 2 minutes, we started seeing a substantial damage and significant kill into the cells. What that tells us, there is a certain dose of plasma, as measured, let's say, by time of exposure, that you have to stay below that threshold before you start causing damage into healthy cells. The epithelial cells, this is how they look like. These are the control. We follow them with time, of course, 4 hours and 8 hours, 24 hours later, so we can compare when we treat them. This is 30-second treatment with plasma, 4 hour, 8 hour, 24 hours, and it doesn't look so bad. It looks like what I showed you in the control. This is 1 minute, and we follow them in time, and it still doesn't look bad. So anyway, I don't want to show you all these, so we put them all, of course, in a plot. And you can see that we can go up to about, let's say, 4 or 5 minutes, and then suddenly they start dying in a major way. And this is a 4-hour post low temperature plasma treatment. So these are the epithelial cells. And I show you here 8 hours post-treatment, 24 hours post-treatment. So basically, if you look at this plot here, so from 0 second to 5 minutes, we have a good viability of these cells after treatment. However, once you hit the 10-minute, you are causing substantial damage. So this is another threshold, which is obviously higher than the fibroblasts. So the fibroblasts are kind of the weakest link, if you like. So if you have to stay below that treatment before you start causing damage to the fibroblast. You can go longer without causing damage to the epithelial cells, but you cannot go indefinitely. And you can find a lot of papers that discuss wound healing where the treatment, lasts up to 20 minutes or so. And to me, that tells me, there is damage that is occurring to the healthy cells. And that's why it is important to have at least some studies on the cell level, in my opinion, before somebody tackles a whole therapy or anything like that, to understand, what are the limitations, because every method and every technology has its limitations. So I showed you now a lot of results, but the plasma cell interaction, what are the burning questions? Back in '96, that I showed you the burning question burned in my mind at the time. Now I still have questions, not so many answers, maybe some results, but not too many answers of what's going on. However, I think our questions have gotten better. Cells are actually surrounded in real application by fluids. So the plasma-liquid interface is very important. Now, all those results I showed you, the plasmas were in suspensions. So they were in liquids because we try to simulate as much as possible that situation. Now, the plasma-generated reactive species in liquid then are important. So we need to identify them and their fluxes in the liquid. What is it that is reaching the cell actually when it's in a fluid, which is much closer to the real situation? And I was talking to Mark this afternoon, and he was telling me about this, the importance of the liquid. Identification and measure of the fluxes are very important. Detailed mechanism of interaction of reactive oxygen species and reactive nitrogen species. We really still, I'm not showing you too many complicated figures that my biologists show me, but a cell is really very intricate, complex system. It's a complex machine. There is a lot of stuff going on in a cell, and the mechanisms that supposedly the reactive oxygen species of plasma, reactive nitrogen, but are initiated into that cell are not well defined. We still don't know too much about that. The intracellular, is the plasma causing intracellular reactive oxygen species to be generated? Because a lot of these can cause cascades of events and signals a release of this factor, of that factor, and there are so many that are important. How am I doing on time, by the way? I don't want to go too long. Okay. When am I supposed to stop?
Oh, pretty soon.
Pretty soon. All right. That, to me, that means 6, 7 o'clock. Okay. So the cell signaling pathways initiated by plasma exposure. We need to elucidate what are these cell signaling, and of course the selectivity. Why is the plasma doing this to this cell, and how about that? How about the cell next to it? It's killing the cancer cell. What is it doing to the healthy cell next to the cancer cell? Right? These are very important questions. And of course, the selectivity between prokaryotes, which are the bacteria, and eukaryote. For example, in wound healing, you are trying to kill the bacteria that is causing the infection, but you don't want to damage the healthy tissue of the wound. Because in reality, this is our jet, these are the bacteria, this is the mammalian cells. What's happening is we are generating this species, this reactive and oxygen and nitrogen species, and they are interacting with this in the fluid. So we really need to know what's happening in the fluid level and what's going on. What are the species that are interacting with the outside of the cell. We know our plasmas are safe. Our jet uses, of course, helium, but what reacts with ambient air. And we have measured, our species, at different location, etc. We know how to do that. The plasma part, we know how to do it. And but the question is, this ROS and RNS, what are doing to the cell morphology? What is the cell signaling they are involved in? And for example, in the case of cancer, what cascades is being activated to tell the cells, hey, it's time to die? Why are they dying? So the question is, what's happening at the molecular level? I showed you that the membranes of, for example, when it comes to bacteria, gram-positive, gram-negative are different. So, of course, we think these species are going into the membrane and doing oxidation to the lipids and to the proteins and disrupting the normal function of those of the membrane. But all this needs to be verified. What we need to do, which we are studying, is the membrane proteins, what happened to them, the metabolic signals. By the way, we did some work on this. I'm not showing it today because I don't have time. The molecular signals, the signal factors, the quorum sensing. Bacteria, for example, when they infect a wound, they actually form a biofilm. And when they are together, bacteria are smart. When they live together, they actually communicate with each other, and they secrete all this polysaccharide film that will protect them, and there is a whole field quorum sensing. So we need to know what plasma is doing to disrupt that communication, of the cell. In case of biofilm, the eukaryotic cell invasion and the biofilm development, etc. All this is very important. This is another one that shows you how the cell, forms a biofilm and then keeps that process going and going. And when they live in a community, they are well protected and they know how to optimize their living condition. Biofilm, sometimes even antibiotics are not effective against them. The effect of low temperature plasma on eukaryotic signal is very important. There's, like I told you, there's a lot of things that happen. They have this cytokine and signal. These are proteins that transmit information in cells. They have growth factors of all kinds. The membrane proteins, the integrin. This is all too much biology for me. But I'm just telling you this slide. I added it because it shows how the complexity of the system, that we need to study proliferation, the morphology, differentiation, protein expression, DNA integrity. All this is really big questions that the field of plasma medicine is faced with, that we need to answer in the near future if we want this to be actually a method or a therapy to be applied in medicine. This is another slide for the apoptosis. Apoptosis, this programmed cell death. There are certain cascades. One of them, this protein called caspase 8 and caspase 3, that can send these signals to the cells and to tell it to self-destroy, basically. And we want to know, are we activating caspase 3, for example? Are we fragmenting the DNA? And you can measure that, because when you fragment DNA, there is this thing get released, and you can measure that. And again, how, what particular protein we are activating. And does plasma reactive species alter these targets? These are all kind of targets that are embedded in the cell. So the approach that we would like to pursue is on 4 stages. One is called the genomic replication, the transcription on the level of the RNA, the proteomic, which is on the protein level, and the phenotypic, which is on the cell function. And we want to really figure out what are all these species that we talked about earlier, and what are their effect on these 4 levels. This is our grand project that should take us a long time to get to the bottom of it. So today, I just wanted to give you at least a feeling of what really the work that needs to be done. The work that has been done is interesting, is showing us a promise, but understanding the science is a different matter. So anyway, to conclude, low temperature plasma offer effective controlled means to inactivate bacteria, possibility of using plasma jet to treat diseases and caused by pathogenic proteins, by killing cancer cells. Low dose of plasma have no cytotoxic effect on fibroblast and epithelial cells. It has to be a low dose. And the mechanism of interaction on the subcellular molecular level remain not well understood. So the prospect, this is how they did it in the Middle Ages, and this is what we still do today. The replacement of a limb because you cannot treat the infection, because gangrene sets in, or what have you, so you have amputations. And the hope is that our plasma here, the plasma technology, which can kill the bacteria causing the infection, but also can heal by causing proliferation of the healthy cells, like fibroblasts. So we call it the plasma kill, plasma heal process. And what's in our toolbox is this reactive species that the plasma generates to cause beneficial effect, hopefully. So I just want to mention that we just published a book on plasma medicine, published by Cambridge University Press, and Mark Kushner here contributed a chapter on plasma chemistry, and I hear it's one of the best chapters in this book. So thank you, Mark, and I would like to thank you, and I'm ready for your questions.
Thank you very much, Munir. Are there any questions? Yes.
So when you were going through the active agents of the plasma, you have temperature, you have nuclear radiation, charged particles, all that. Do you think that there's any factor in like the blunt trauma of impacting these, bacteria and these cells with this high pressure, very fast propagating bullet? Like, is there any?
No. The bullet. By the way, we don't to kill this bacteria. We killed it without the bullet. In a normal, just a plasma between two electrodes, and we put the sample there, and we killed them with the bullet. So this is not really a physical effect. This is not, a force. As I mentioned, the bullet is actually not a propagating a solid piece of material that is propagating. It's actually an ionization wave. It's so you're not like hitting them. You are imagining that bullet like a bullet coming out of a gun, with a big impact. So the bullet is actually more like a vehicle that transports some of these, electric field and reactive species and things like that. So this is not a trauma by collusion or physical effect. It's not.
Why would there be a selectivity between, bacteria and mammalian cells?
Okay.
If the process that you described was the electrons attaching themselves to the cell and then electrostatically pulling it apart.
No, well, that's not what's happening in all cases. I wanted to show you one case where I don't talk about the reactive oxygen species, reactive nitrogen species. That charged species could play a role. The structure of mammalian cells and of bacteria of prokaryote bacteria is very different. It's extremely different. For example, in mammalian cells, all the DNA and other parts of the cell, they're all contained in organelles. They have their own walls, their own membranes. They are well protected, and mammalian cells have I guess through evolution developed a way to, because, for example, we have reactive species. We have radicals that our own body generate all the time, and our cells have developed method and means to cope with that, to negate the effect of this reactive species. In bacteria, for example, the DNA is floating in the cytoplasm. It's not contained in a nucleus. It's not like the eukaryotes where the DNA is in the nucleus. And so if you have a way to disrupt the outer membrane of a bacteria, a lot of the contents that life depends on, like DNA and other things, can just leak out. And the bacteria turn out to be a lot more susceptible to be affected by plasma than mammalian cells. And I'm sure there are other things that I might not be able to tell you exactly. Yeah.
Have there been any work on because you are talking about bacteria? What about viruses?
Oh yeah, I didn't cover that. Yes, so people have done work on viruses and fungi and all that. They're very effective.
Okay.
Yeah.
So in your mammalian studies and the post-exposure experiments, have you ever figured out like how long you can do repeated exposure? Like, for example, you said or in the results you showed after 5 minutes, before 5 minutes, it's still going to be normal. But how about repeated exposure? I was wondering.
Yes. We haven't done that, but I know of people who have done that. Who, for example, there are people who actually did this wound healing work, and what they do is actually they do a regimen, right, where it's exposed, let's say, over a month time, and every day or every other day for like 10 minutes or 20 minutes, and they keep repeating that, and that showed good results. Like I said, there are pictures that show that. I never show them because it's not really my work. But there are people who have done repeated exposure that showed benefit. Yeah.
We have various plasma devices, and we use them to treat various bacteria, and all the effects show some plasma can do something with the bacteria.
Yeah.
But how to extract like the scientific process behind the plasma, behind the interaction between the bacteria? Do you think it is necessary to have like a standard device, and we put some effort to do some diagnostics and extract all the reaction mechanisms?
You know, this is a good question, and it's been debated in our community to have, because everybody has his own device. Everybody is doing their own, thing, and there is no really standard. It's like the GEC cell or something like that, where everybody has the same device working under the same condition, so that we can compare our results. And that has not happened. So we are actually, everybody's doing his own thing, and we meet, every year once or twice, and we show our result. But you are very correct. Within the community, in order to really compare results, compare apple to apple and orange to orange, that standardization needs to be done. I agree.
Michael?
Talk about killing the cancer cells, and I was wondering about like delivery mechanism, and particularly for like if you have a way of more widespread of cancer, like it's more widespread throughout your body than maybe some of the localized like cancerous tissue. There, a mechanism to think of to.
You mean a cancer that metastasized all over the body? Yes, and what's?
Do you think that the plasma could be useful in that sense for those kind of
I
things?
don't know. This is. I would say no, but I'm not a doctor. I just play one on TV. So I don't know because once it's metastasized, from what I know, it's very hard. Even chemotherapy and all that is. But I can see a combination of therapies. It doesn't have to be just plasma. For example, for the leukemia cells, we know we can kill the white blood cells, but the leukemia is overproduction. You kill them, but they can keep coming back, right? So you have to actually treat the root of the problem, which is probably the bone marrow. So I can see, for example, the plasma be a part of a therapy, not the entire therapy. Because there are, combination of therapies. Actually, sometimes you hear in the news that some really medical challenge that can be met with a combination of therapies, not just one therapy. So I can imagine the plasma playing some role, but I have a feeling, for if you are not treating, if you're treating something that has metastasized all over a human body, I don't know how to answer that.
Thank you very much for the presentation.
Thank you.